When you hear the word cancer, your mind probably jumps straight to our genes, right? To our DNA. But what if I told you the fight against cancer isn't just about fixing broken hardware? What if it's actually about debugging the software that runs our cells? Let's dive into this totally revolutionary way of thinking about cancer.
For decades, the story we've been told about cancer is that it's all about genetic mutations. A typo in the code of our DNA that makes cells go haywire and grow out of control. And look, that's a huge piece of the puzzle, for sure, but it's not the whole story.
And this idea right here is the new frontier in cancer research. Think about it like this. Your DNA is the computer's hardware. It's the physical blueprint, the circuits. But the software, the operating system that tells that hardware what to do and when to do it, well, that can get bugs too. In our bodies, that software is called the epigenome. So, what is this cellular software? This is where we get into the absolutely fascinating field of epigenetics. It's this whole other layer of instructions that sits right on top of our DNA, conducting the orchestra of our genes.
Okay, so here's the crucial point. You got to get this. The DNA code itself, the hardware, doesn't change one bit. Epigenetics is all about controlling which genes are being used and which ones are being ignored. It's kind of like taking a blueprint and adding sticky notes and highlights all over it, telling the construction crew, hey, build this part, or nope, ignore that section for now.
In a healthy cell, the software just hums along. Everything runs smoothly. But in cancer, that software can get corrupted, a virus if you will. This rogue code can literally tell the cell to shut down its own defense systems. We call those tumor suppressor genes. And at the same time, it can activate the very genes that pour fuel on the fire of uncontrolled growth. So let's look at how it actually pulls this off.
First up, we've got DNA methylation. Imagine a tiny chemical tag getting stuck directly onto a gene. This little tag basically acts like a big, red do not read sign. It just stops that gene from being used. In cancer, these stop signs can get plastered all over the good genes, the ones that are supposed to protect us, effectively silencing them.
Next, there's histone modification. See, our DNA isn't just a loose strand. It's wound up super tightly around these little proteins called histones, like thread on a spool. Epigenetic changes can either loosen that thread, making a gene easy to read, or it can tighten it up, hiding the gene so it can't be used. Cancer can hijack this to hide the good stuff and put all the bad genes out on display.
The third mechanism is even more incredible: chromatin remodeling. Here, the cell's own machinery literally moves those spools of DNA around. It physically remodels the architecture to either show off or completely bury entire neighborhoods of genes.
And finally, we have non-coding RNAs. Don't let the name fool you. These things are small but mighty. They're these little molecules that zip around and act like molecular kill switches. They can find a specific gene and just block it from being used, flipping it into the off position. Okay, so we have these 4 incredibly complex layers of software running our cells, which leads to the obvious question: how on earth do scientists read this ridiculously complicated code to find the bugs? Well, the answer is a field with a cool name: bioinformatics.
The scale of this challenge is it's staggering. We're not talking about a few lines of code here. A single person's epigenome generates trillions of data points. It's an amount of information that is so vast, no human brain could ever possibly sort through it alone.
And this is where bioinformatics comes to the rescue. It's basically using supercomputers and incredibly smart algorithms to manage, analyze, and make sense of these massive datasets. It lets researchers see the patterns in the noise, find the glitches in the software, and link those specific glitches directly to cancer.
But just looking at the epigenome isn't quite enough. To get the full, high-definition picture of what's going wrong inside a cancer cell, scientists have to pull in information from a bunch of different levels. They call this a multi-omics approach.
Let's just quickly break this down using our computer analogy. Genomics is the hardware, right? The raw blueprint. Epigenomics, as we've said, is the software, the notes on that blueprint. Then you have transcriptomics, which shows us which parts of the blueprint are actually being copied and sent to the factory floor. And finally, proteomics shows us the actual things, the structures, being built from those copies. You need to look at all 4 together to really understand what's happening. So, we can read the faulty code. What's the payoff? Why does this all matter? Well, this is where it gets really exciting. Because if cancer is a software problem, then maybe, just maybe, we can create therapies that act like a software patch.
And this right here, this is the absolute game changer.
Genetic mutations are permanent changes to the hardware. They are incredibly hard to fix. But epigenetic changes? They're just software. They are dynamic, and most importantly, they are reversible. This opens up an amazing therapeutic opportunity. We don't have to replace the hardware, we just have to rewrite the corrupted code.
And the strategy is actually pretty brilliant in its simplicity. Step 1. Use bioinformatics to find the exact epigenetic error that's driving a person's cancer. Step 2. Create drugs that go in and block the very enzymes that are writing that bad code. And Step 3. By doing that, you restore the normal gene patterns. You basically reboot the cell's operating system back to its healthy factory settings.
Now, of course, it's not that easy. I mean, if it were, we'd have it all solved. Researchers are up against some major hurdles. How do you make sure your software patch only fixes the cancer cells and doesn't mess with your healthy cells? What do you do when cancer cells get smart and build firewalls to block the drugs? It's a massive, ongoing challenge. But even with those challenges, the progress being made is just undeniable. This entire approach is reshaping the future of cancer treatment, pushing us towards something way more precise and personalized. And if you look out to the horizon, you can see what's coming. We're talking about true personalized medicine, where your treatment is designed specifically for your unique epigenetic signature. Then there's the even more futuristic stuff, like epigenetic editing, using tools like CRISPR to go in and be a molecular programmer, literally debugging the code. And a whole new class of drugs is emerging to control those little RNA switches with incredible precision.
And this isn't science fiction. The goal here, as the researchers themselves put it, is to completely redefine the therapeutic landscape for cancer. It's a shift from using sledgehammers to using scalpels, or maybe even a keyboard and mouse.
Which leaves us with this one final provocative thought. We update the software on our phones all the time to fix bugs and improve how they work. So what if the next great leap in cancer therapy isn't some new chemical at all, but just a simple, elegant software update for our very own cells?